I was just thinking, based on that conversation we just had, that if I were to donate my body to science, they'd probably just return it.
Uh,
No, thank you.
no thanks.
We've seen better.
Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Jordan Ruggieri.
And I'm Cassie McCreary. And today we take Absolute Genius overseas for a fabulous conversation with Dr. Raquel Muñoz of Sevilla, Spain.
Raquel recently finished her doctoral studies and works as an immunologist on the forefront of innovative cancer research. She's opened our minds to what the future of cancer research looks like, and we're excited to share our conversation with you today.
How exciting for us that we've gone international.
I know, it's so cool to have our first international guest on the podcast. I think it's absolutely amazing.
Some might say it's absolutely genius.
We're awful. I use absolutely in everything now. I can't get away from it.
Can't stop, won't stop.
Raquel, thank you very much for joining us today on the Absolute Genius podcast. We're really excited to have you with us and to talk about your research.
Hi there, me too. Thank you for inviting me.
Can you give us a background of what your research is and who you are for our listeners?
Yeah, my name is Raquel. I work at Hospital Universitario Virgen del Rocío in Sevilla, in Spain. And our research is about CAR T cell therapy. Have you heard about it?
Yes, yeah, CAR T is awesome. Can you talk a little bit about what CAR T is for the listeners as well?
It's a type of immunotherapy. We can use the immune system to protect from an illness. So we can modify the immune system and for the treatment of this illness. And for example, with the CAR T cell therapy, we obtain T cells that are immune cells. These are the fighters of the immune system. We can modify them in the lab and put a specific receptor that can find and kill cancer cells. So we create the CAR T cell, that is the acronym of chimeric antigen receptor T cells, because we have added this chimeric receptor in the cell surface. So when we create these CAR T cells, we can reintroduce them into a subject, and the CAR T cells will find and kill the cancer cells in a specific way.
That is incredible. So when you are working with CAR T, can you describe a little bit of how you know a modification was made properly?
Yeah, we have markers gene, so we try to amplify these marker genes. So we know that the CAR T cells that we have made have a specific gene. So if we amplify this gene, we know that we have done the CAR T cell well. And if we don't see any amplification, we have failed with the construction of the CAR T cell.
Do the CAR T cells stay a long time in a subject, or do they degrade over time?
We are researching about this because it's important to know how many days or even months the CAR T cells still and do the work, because we want that they work only in a period of time, because if they still killing, maybe we can have side effects. We don't want to have it. Our main that we want to achieve is to measure how many time these cells is still in the blood. So we try to measure and describe what is the curve of the expansion of these CAR T cells.
Very interesting. What are you hoping the ultimate outcome is associated with your work?
We want to achieve to monitoring the expansion curve of the CAR T cells. And until now, we have described 3 groups. The first group that we have described, it's a group that expand the product that we have infused between day 10 and 11. The second group, it's a late expansion group, and they expand the product after day 11. And there is a third group that have no expansion. So we are trying to compare if there are any difference between these 3 groups. And we have seen in a preliminary research that maybe it could be any difference in terms of side effects or failure of the CAR T cell therapy. So our mark is is trying to prevent and predict the outcome of the therapy.
You talk about expansion. Can you elaborate on what do you mean by expansion? Is that growth of the modified cell line within a subject?
Yeah, we talk about the increase of levels of CAR T cell that we can detect on the blood, because there is an increase first and then a decrease. So we don't know, and this is what we want to know, that if this increase and decrease is related to the effects of the CAR T cell, and if the amount of the CAR T cell that we can that we can detect in the blood is related to the effect of the therapy and the side effects also.
Interesting. So you are trying to understand what happens when you put the CAR T cell, the modified cells, into the subject to determine what the cells grow, and then the cells and reproduce, and then the cells decrease, and how that impacts the subject and outcomes of that CAR T therapy. Is that accurate?
Yeah, because maybe if we understand this curve of expansion, maybe we can predict the outcome. And for example, if we don't see an expansion, maybe we could give a second dose of CAR T, for example. And maybe if we see early expansion or of a high amount of CAR T cells, maybe it could be related to side effects, and maybe we could give some drugs to prevent these side effects that could be fatal. For example, we talk about neurotoxicity or cytokine storm. So if we can prevent this, maybe the outcome it will be better.
Raquel, what technologies do you use in your research? Do you run NGS or digital PCR?
Yeah, we use digital PCR, in fact. We tried to measure the expansion of CAR T cell first with real-time PCR, but we had a lot of problems because, as you know, with the real-time PCR, we need to amplify a reference target. And in the same reaction, we have all the reagents and the primers to amplify our target, but also the reference target, sorry. So in cases of low frequency targets, as our case, there is a high probability of competition between the reagents. And maybe we could amplify only the reference target because there is a high proportion if we compare with our target. So this problem doesn't exist with digital PCR because thanks to partition, we have micro amplifications. So we do an absolute quantification. We don't need a reference. So we obtain an absolute quantification, and in our research, this is a great advantage. So we decide to use digital PCR.
Awesome. So digital PCR, instead of comparing to a standard curve or to a reference, you're able to take that copies per microliter number and use that to plot your expansion curve. Is that accurate?
Yeah, this was the clue of our research, because with real-time PCR, in some cases we wasn't able to amplify our target because there was very low proportions.
Oh, I see. So your limit of detection is much lower when it comes to digital PCR. Your limit of quantification is lower, so you're able to see those smaller changes easier. Is that accurate?
Yeah, and that's a great advantage for us, because with digital PCR, we are sure that if we don't see amplification, we are in the third group that as I said before. But maybe with real-time PCR, we are not sure that if we are in the third group and there is no amplification, there is no expansion, or maybe there are competition between the reference and the target. So using digital PCR, we are sure about this.
That's great. Do you still use reference genes when you use digital PCR to quantitate? And do you use a smaller number of them compared to qPCR?
Yeah, in digital PCR, we use a reference because we want to know that if we don't see amplification of our target, but we see amplification of the reference target, we know that there are cells, but they are not CAR T cells. And also, we use the reference to normalize different measures of different days, because maybe in different days we have different amount of cells. So we use it to normalize.
Makes sense. So you use a reference to make sure that the amplification process is working. So if you end up with, let's say, 0, right, no amplification of the target you're looking for, you know that the process still worked and it's a true 0. And then you also use that reference to normalize and make sure it's an equivalent quantification across multiple days.
Yeah.
We talked a lot about digital PCR and even qPCR. Do you use any other methods, any cell culturing or next generation sequencing, or what does a day in the lab look like for you?
Yeah, it depends on the area. For example, in histocompatibility, we use a lot of molecular biology like NGS. In autoimmunity, we use more cell culture and IFI. It's the acronym of immunofluorescence, indirect immunofluorescence. Yeah. And in immunodeficiency, we use a lot of flow cytometry.
Interesting. A lot of different techniques. I forgot a question here. I want to go back a little bit. When you are working with the CAR T cells and the process, how do you modify those cells? So do you use CRISPR-Cas9 as part of your modification workflow?
Yeah, This part has been made by my colleagues of hematology, not for me. But yeah, they use this technology. Yeah.
Can you elaborate or even just from your knowledge on what that might entail when making modifications to the cells?
Yeah. We obtain the T cells and cultivate them, and we try to expand them in the cultive. And then we use this technology that we have talked about, and we put the gene that codes the chimeric antigen receptor. So the T cell will express this receptor in the cell surface. So now we have created the CAR T cell. We will expand these CAR T cells, and then we can use them.
That's incredible. Amazing research there.
Yeah, I think it's the future of the cancer treatment.
Are CAR T therapies more common now, or are they working only for particular sets of illness?
The CAR T therapy, it's more studied in lymphomas and leukemias because the cancer cells are in the blood. But I know that nowadays there are a lot of research that are trying to use CAR T cells in solid tumors, and the results are good. I'm sure that we are going to hear about it more and more.
Taking a quick break from our conversation to tell you about Applied Biosystems QuantStudio Absolute Q dPCR system. This instrument enables quantification of your targets without the need for standard curves in only 90 minutes. Digital PCR can be as simple as preparing your samples, loading onto the plate, and running the instrument.
Unlike other digital PCR systems, the Absolute Q dPCR instrument does not use emulsion or other droplet-based methods to compartmentalize reactions. In fact, the microfluidic array plate technology enables consistent delivery of more than 20,000 microchambers. It's a great solution for anyone looking to quantify gene targets.
And Thermo Fisher Scientific has a suite of dPCR assays for applications like AAV viral titer quantification, liquid biopsy analysis, and wastewater surveillance. You can learn more at www.thermofisher.com/absoluteq or visit the Absolute Genius webpage. Again, that's www.thermofisher.com/absoluteq or visit the Absolute Genius webpage.
The Applied Biosystems QuantStudio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures. Let's get back to our conversation.
Bienvenidos al Cassie's Career Corner. I don't know how to say corner in Spanish. So this is the point where I will ask you a little bit about your studies, like academic studies, your education, your career path, a little bit about kind of advice for students and that type of thing. So can you give us, please, an overview of your academic studies? Because you graduated relatively recently.
Yeah, I studied biomedicine, and then I did a master of clinical diagnosis and lab diagnosis. In my work practice at the master, I realized that I loved hospitals and lab hospitals. So I decided to specialize in immunology. So I had to do an exam to do the specialization because it's the system here in Spain, and yeah, and I passed this exam. Yeah. And I could choose between different specialization, and I choose immunology. And I have studied different areas of immunology for 4 years. And now I'm officially immunologist.
Yay!
And I have been working at a hospital as immunologist for 2 months in Barcelona. And now I'm trying to finish my doctoral thesis.
So how did you decide? Because at least the way that it would work over here in the US, if following a route like yours, a person would probably have to decide between going down PhD studies and research or medical school. Sometimes people do both, and you can do an MD, PhD. How did you decide you wanted to go more down the research route versus like medical school, for example?
Yeah, I don't know if I decide.
Okay.
Yeah, I only
It's never too late, so.
I loved research and know more about human physiology and human illness. So any project that I could be in, I tried.
Great. What is it about immunology that you like so much?
I think it's an area that you can learn about any illness or any physiology process, because immunology is related to anything. Immunology is related to cardiology, to neurology, gastrointestinal, nephrology. So I think it's a complete way of know more about human physiology and illness.
Yeah, it has a wide range. So it keeps things very interesting, I would imagine, to study, right? Because there's so many different, I guess, applications. For somebody who's maybe just starting out, either in their career or towards earning a PhD or advanced studies, what is a piece of advice you would give them?
Maybe I will say them that ask people for their jobs, because I think I didn't know very well what people do in their jobs. For example, I didn't know that a doctor could be also immunology. I thought that doctor is only about surgery, or so. I would say them that they have to ask and make their own opinion about different jobs and degrees and careers, so they can make a good decision if they know what people do in their jobs.
I think it's probably natural for a lot of people at one point or another to feel a little bit of maybe self-doubt during their research or question if it's something they want to continue. Did you ever experience something like that? And if so, how did you move past that?
Yeah, of course. The research is very hard because you have many questions, and you have to try different things to try to answer these questions. And more the great majority of these things that you try, they fail. And these are also results, but when you are trying to do things and you fail, it's very hard. And I think it's frustrating. But when you have a result, and you see that all the work that you have done, you could express, and you could write an article, or show your results to your colleagues, it is also very very happy for you. So I think there are mm, good things and not as good things, but the research, it's very it's amazing.
It sounds like having a very good support system is really important in those times. Having colleagues who will support you and other students and your mentor and all that. And I think you hit on an important point, which is I think people forget that a lot of science is built on failure just as much as it is success. People tend to just forget about the failures and move past and not cover that and always glorify only the success. But a lot of times it took a big degree of failure to get to that success, right?
Yeah, I think that to have a positive result, you have to have 100 of negative results.
Right. In your time in the lab, for you, what was your most exciting or proudest moment?
I think my proudest moment was the first time that I saw my name in an article as an author. It was the description of a new allele or something like that. It wasn't an very important article, but it was my first article.
Yeah.
And it was amazing.
Yay. Okay, how about something, you don't have to share it if you don't want to, but how about something that was maybe like a mistake that you made in the lab, or something funny or embarrassing that might have happened sometime? Jordan has a good one if you need one. So
I think I have handled this.
Okay. All right.
I have one that is embarrassing, but also funny. When I was in my first week, I think, yeah, in the lab, in the hospital lab, I was looking for some samples in the fridge, but in a room that is a fridge. It's a room, but the temperature is under 20 degrees. So I was looking for my samples, and suddenly the door was locked.
Oh no.
Yeah,
no.
and I thought, keep calm, get your phone and phone a colleague. He will open the door. I have no phone.
Oh no.
So I started shouting out loud because I thought I would die,
Yeah,
frozen.
right. Oh no.
Yeah, I was shouting out loud for 10 minutes, I think. And then a colleague opened the door and said, "Are you crazy? This red button, if you press and if you push the button, the door opens."
Oh no. Like an emergency button.
Yeah.
Oh no.
It's a security system.
Yeah.
It makes a lot of sense
Yeah.
because it's a fridge. But I didn't know. And I think all the lab was laughing about me for
Oh no.
days. Yeah. But I was very happy for not dying frozen in my
Yes.
first week.
Yeah. Oh, that's a good one.
I don't know if people can, I don't know, understand how many mistakes you make in that first week or so. I made so many funny mistakes my very first week in the lab.
That's how you learn.
It's how you learn. Yeah.
Because they're never going to cover that in school. You have to mess up. That's how you learn. And everybody needs a little bit of a break from work and research and all of that. What do you enjoy outside of work?
I enjoy a lot doing pole dance. Do you know what it is?
A pole dancing?
Yeah.
Raquel, that's
Pole dance.
awesome. That's great exercise, I've heard.
Yeah. Yeah. I'm strong.
Strong. Yes. Okay, excellent. That is so
Yeah,
funny.
I think it's the only hours that I don't think in the job or any problem, or yeah, because you have to be very concentrated, because if you are in the pole and you
Yeah, you fall off.
yeah, it's dangerous. It's dangerous.
I believe it.
My sister does the silk dancing from the silks that hang from the ceiling.
Oh, like the aerial
And
acrobatics or
the
whatever.
aerial,
Yeah.
yeah. That's I don't know how anybody does the poles or the silks. I can barely hold myself up for 3 seconds. So,
Oh, that's so funny. So, Yeah, it's important to take a break and get your mind off of work. Otherwise, you'll run out of ideas and you'll get burnt out, right? So, it's- Jordan, now that's all I can picture is you like just falling off of a pole. It's like, Raquel, what what would you like to see out of your career in the next maybe 5 years or 10 years?
Uh, yeah, it's a great question.
You don't have to know. It's
Yeah,
okay.
yeah. I don't know. The only thing that I want is finish my doctoral thesis, and yeah, I wish in 5 years
You
it will
will.
be finished. Yeah,
You will.
yeah, for my mental health.
Yes. That's so funny. All right, great.
Well, Raquel, thank you so much for joining Absolute Genius today. You are our very first international guest, believe it or not. So we are very excited to have you on and to have you be a part of this. So thank you very much for your time.
Thank you very much. I was very confident because of you. Thank you.
That was Dr. Raquel Muñoz at Hospital Universitario Virgen del Rocío in Sevilla, Spain. Thank you so much for joining us on today's episode of Absolute Genius. It was produced by Sarah Berganti, Matt Ferris, and Matthew Stock. Stay curious, and we'll see you next time.
By the power vested in us, we are geniuses. Yeah, we're gonna have to be self-proclaimed because there's not a single institution that would sign off on that statement.
It's the institution of a home printer. That's what it's called.
Did you know, speaking of home printers, I am ordained?
Oh.
I should do the same thing, but genius.